Okay, so here we go with the nervous system and our study of the nervous system. So, we got, to me, it's always been a little interesting the way that people have put this, where they put this, because when you think about what system keeps you in all homeostasis, the nervous system, isn't it? And so you would almost think, well, shouldn't we start with that? And your textbook doesn't, and I don't know. I might think about changing that sometime, the sequence of that. But anyway, go ahead and follow our textbook with this. So the nervous system, we already know a little bit about it, don't we? We certainly know that it is the organ system that controls all of homeostasis in the body, and it does that because it's a special kind of tissue. The nervous system is composed of special kinds of cells that make up tissues that are able to detect signals in the environment, internal and external, and then communicate with other cells. We just saw in the muscle system how motor neurons communicate with skeletal muscle, didn't we? We saw that. But we have also been introduced to the idea that the nervous system communicates with endocrine glands. Endocrine glands are epithelial tissue that secrete what? Hormones. So the nervous system can communicate. It's like the great communicator, right? It communicates with any of these other systems, trying to keep the person in a state of balance, a state of well-being, a state of homeostasis. And it mostly carries out these functions by negative feedback. Any questions about negative feedback? No, because we've learned about that, right? You guys in histology have also learned in histology that there are two major types of cells in the nervous system. They are the neurons, which are these, large cells that have these structures coming off of them like this. We saw motor neurons, didn't we? And then there are those supportive helper. They're called helper nerve cells, and those were called glial cells, also known as neuroglial cells. You'll see them referred to as both. What is a glioma? If you heard that somebody had a glioma, what would that mean? A tumor of the what cell? Of one of these glial cells, right? Okay, so fine. So we have a little bit that we're building on here, and we're going to take a closer look at this nervous system. It is working hand in hand with the endocrine and the muscles to help to keep us in homeostasis. We do know that. When we're studying it, we have these divisions that we learn about. We learn about the central nervous system and the peripheral nervous system. You would want to know that the central nervous system includes the brain and the spinal cord. The brain and the spinal cord. And then the peripheral, to the periphery, to the side, right? The peripheral nervous system are going to be those nerves that come off of the brain and the spinal cord. Fair enough? Now, when we think about that, does the anatomy of where the brain and the spinal cord, is it different than the anatomy of the peripheral nerves? It is. They're completely encased in bone, aren't they? The central nervous system, they're completely encased in bone. And we're going to find out that they have 3 separate membranes that are isolating and compartmentalizing them. So we didn't just, scientists didn't just do these divisions to make you learn something else, It's because these tissues are very uniquely different from each other. And we have suggested that if damage gets done to the central nervous system, neurons, do we expect healing the way that we could potentially get with peripheral nerves? Yeah, so it's a difference in actually the actual cells that make up the tissue, isn't it? So that's going to be important to know. I would actually want you to know that when we think about these divisions, and here's a picture of them where you have like flesh, this whatever color for the brain and spinal cord, and then you have these peripheral nerves coming off, that there are 12 pairs of cranial nerves that we're going to learn. We're going to learn those. We're going to learn those, all of them. but there are also 31 pair of spinal nerves that are coming off the web. Spinal column. So, and again, where they're yellow here on this picture, those are representing the peripheral nervous system. Fair? All right. Now, when we think about this peripheral nervous system that we said is kind of like web-like when you think about these nerves and how they're going out all to the fingertips, to the toes, right? You know, all the way. We know that there's two divisions there. There's the sensory, and then there's the motor or efferent system of the peripheral nervous system. So what do you think the sensory does? Senses, right? And it's sending, so this is it's called afferent because it's sending to the central nervous system. The central nervous system, which is the brain and spinal cord, will integrate those signals, which means that it's going to figure out what message is being sent. And then the central nervous system is going to send through the motor or efferent division a response to that signal. So the reason I'm getting you guys to know that is because of this. There's some diseases that attack just the motor. There's some diseases that attack just the sensory. You get what I mean? And you've got to have them all working. And you've got to know which ones do which, because that's going to help you to understand why your patient has lost what they lost, right? So anyway, within this motor division, the efferent, visceral, if we say visceral, what is that referring to? What do y'all know that's kind of referring to? Organs, isn't it? It's really referring to like the visceral organs. When you think about visceral organs, that's what you're referring to. Somatic, the somatic is referring to mostly like skeletal muscle, whatever. But okay, that's fine. Now, those of you who like this kind of organizational thing, this would be also a good thing for you to look at, just to kind of see. Now, I want you to know the difference between what's called the sympathetic nervous system and the parasympathetic nervous system. You ready? And this, we're looking at this, and this is affecting the visceral organs, right? I want you to know that this is part of your autonomic nervous system. And I want you to know that when you hear about the sympathetic nervous system, you already have a name for it. You call it your fight or flight response. You call the sympathetic nervous system your fight or flight response. In a fight or flight response, does it take you long to get jacked up to be able to survive, to stay and fight, or to what? Run. That's exactly right. Does it take you very long? No, it doesn't. And so it actually takes a microsecond. If we were to hear something that would be alarming, or to see something that would be alarming, or to smell something that would be alarming, it does not take long. What happens to your heart rate? Increases. What happens to the contraction strength of the beat? It's stronger. What happens? You can feel that, but you don't really feel this next part. But what do you think happens to your blood vessels? That smooth muscle, what does it do? They clamp down, don't they? So blood pressure goes up, heart rate goes up, heart contraction strength goes up, respirations go up. This is because you're going to be willing, you're going to be ready to stay and fight, or you're going to be willing to run. You're going to be needing to do one or the other. It can actually be so exaggerated on some people that they freeze. They wouldn't have made it if a bull was chasing them. That's right. That would have happened. So anyway, no. So that's the sympathetic nervous system, because some people do freeze. That's right. The parasympathetic system is the system that should be going the rest of the time. When the sympathetic nervous system is not engaged, the parasympathetic system is the one that you want going. This is the system that some people call your housekeeping system. This is the system that is going to allow for us to digest our meals that we eat. Keep blood flowing to that whole 30-foot digestive tract. It's going to help with tissue repair. Your immune system is going to be part of this. Hey, look, if you're being threatened by something, do you need a lot of energy going to your immune system that's going to help you to survive the influenza virus that you encounter next week? No. If you've got something threatening you, do you need to digest that meal that's going to help you to build, whatever tissues? No, because you might not ever get another meal. So you got to get through that first, right? How many of you are stress eaters? So is that even a smart thing to be doing, stress eating? Because if you're stressed, is your body even handling the nutrients you're putting in? Is your body even moving those substances along at the pace it should have been moving them along? Is your body even able to even address it? No. So it's just really whatever. So these are that. Now, we're good so far? See if this doesn't kind of make sense to you. This is the text of what this picture shows. If I were to touch something that's hot, that's the sensory peripheral nerves. I felt that it was hot. That information has to go back to the inner neurons. Sometimes they're just reflexes, so it doesn't even have to go very far. It can loop around. But then what am I going to do? It's going to go to the motor division to move away, right? That makes sense, doesn't it? So as I said, there's some diseases that attack one and or the other, some both. But we're going to talk about some of those diseases, right? So if you've got a patient who's completely paralyzed, and you might someday have patients who are completely paralyzed for whatever reasons, trauma or diseases like ALS, amyotrophic lateral sclerosis, which is what people call Lou Gehrig's disease, or for whatever reasons, they're completely Guillain-Barré syndrome, which is a temporary kind of paralysis, but they're completely paralyzed, they're not moving. And they may not even be able to breathe, so you might be helping them to even breathe. But does that mean, and they can't communicate with you if they can't be doing this stuff, but does that mean they don't feel every time you take their blood pressure or give them an injection or do a Foley cath or bathe them? Does that mean they can't feel? They can feel, because it wasn't the sensory that was affected, it was the motor, right? Okay, so just so you kind of know. All right, now, what we said about neurons, it's so cool, and they get all the glory, don't they, of the nervous system. These neurons get all the glory because they're the ones detecting, and they're the ones that signals are being sent through. And some of them are more than a meter long. Some of their little appendages are more than a meter long, and they're able to send those signals to the next cell. Sometimes the next cell is another neuron, like here. Sometimes the next cell is another neuron. And sometimes we said that with the motor neurons, sometimes that next cell is going to be muscle and or an endocrine gland, right? But we know that they're the great communicators. And when we think about these, this is a typical looking neuron. I do want you to know that the dendritic fields, the dendritic regions, fingers, I call the dendrites, these are called dendrites. These dendrites, which means when I refer to them as dendritic, that's what they are. These dendrites are the feelers. They're the feelers, okay? So they're where the action potentials are being generated is here, these dendritic fields. Now look, when I say action potential, what do I mean? What is happening here at these dendrites? What's happening here? Something's happening to cause what to happen on the membrane? A receptor to open, and what pours in? Sodium. And what pours out? And what happens to the membrane? A shock, electrifying, and that's going to move through the cell. That's the action. It's no different than what happened in that muscle fiber. An action potential is always that. Is always that. And if this is happening at the sensors are going to be these dendritic areas for this neuron. And that electrical current will be sent down. Now it's going to be sent down through a structure known as an axon that is going to meet to the next cell. So guys, you will hear fancy things in textbooks where that's referring to the presynaptic cell and the postsynaptic cell, right? But what does that simply mean? If it's the presynaptic cell, then it's what cell detected, right? That's the cell before the synapse. And the postsynaptic cell is the one after the synapse. It's getting the signal. Are we fair? Is that fair? Is everybody getting that? So the presynaptic cell, postsynaptic cell, that's all that's referring to. Do we need the neurons, especially if even, let's say they're in the brain and neurons are talking to other neurons. Do we need the presynaptic cell getting the signal? Do we need the synapse to be clear, for there to be a clear space so that the neurotransmitter can cross that space? Do we need that to be a clear space? Yes, so that the postsynaptic cell can get its signal. Is that right? And then the postsynaptic cell, an action potential gets generated and it can send it to another cell, Are you with me on this? Think about these cellular pathways that have to happen. Think about those cellular pathways that have to happen for you to, how many of you have ever smelled something and it took you back to a memory? Is that, how long did it take that to happen? Just like that. Is it like it gives me chill bumps? Because that's happened to me one time. That was just like I just couldn't even handle it. Right? Is that the way it happened to you, Maria? You can almost tell from her face the way she's thinking about it. And it took us just not even a second, didn't it? For that to happen, though, it took these specialized. Don't write this down. But think about how amazing that is. It took these specialized cranial nerves that are specialized for smell. They had to send that signal to quite a few other cells, and in your brain, a very special area in your brain where you store memories. And then they got those signals, and then all of a sudden in your mind, you had emotional feelings, visions of the memory. You had all of that happening, and it happened in a microsecond. It's crazy. It's happening at a cellular level. Life is at a cellular level. How things we take for granted are happening at a cellular level. When things go wrong, at what level is it at? That was, you don't end with at. I just said, but where was it? At a cellular level. Is that right? Okay. So that's why we're here where we're here. And I hope that you guys are kind of getting that. Now, let me ask you something. This is like FYI for you. If we can do all these things, if we can tickle my hand, I won't be looking. Okay, I felt it when she did it. No, I didn't. Okay, now that to feel the pressure of that would take a very different kind of neuron sensing than one that if somebody had poured cold water on my hand and I wasn't looking, but I would still be able to say, oh, cold water versus somebody tickling versus something else, pain. So does it make sense to you that the different neuron, it would require different anatomy of neurons to pick up pain versus temperature differences versus pressure? These are called mechanoreceptors. Pressure. Would it make sense to you that their anatomy looks a little different depending on what their function is? Because what dictates function? Structure dictates function, doesn't it? So this is all FYI, but all of these are what? Neurons. They just have different modalities. They have different functions, okay? So I would hope that kind of, and I'm not going to ask you, but I would hope that would make sense, that they have different jobs. And because of that, does it make sense to you that some people are actually have problems with particular ones? Like some people can't detect pain, but is pain an important thing for us to be able to detect? It absolutely is. So where's the problem? In all of their neurons, their sensory neurons, or just the what? Yeah, the pain ones. Or you get what I mean? Right. So that's all that's about. Now, here's what I'd like to introduce to you, and I'd like to do this with a picture because I'm a visual learner, so I make y'all suffer that way. But I do want to do it with this picture. So we said the central nervous system is composed of what? What's the central nervous system composed of? Brain. Wait a minute. Brain. Sorry, that was weird. Brain and spinal cord, right? And that's true. And the peripheral nervous system, we said, are 12 pairs of what? And 31 pairs of, okay. And we know that of those cranial and spinal nerves, some of them are sensory, and some of them are what? Motor. Okay. So we're building our information about this. I want you to know that there are 6 glial cells. Four of them are found in the central nervous system. Where would that be? Brain. And 2 of them are found what? In the peripheral nervous system. So I'm going to talk about the central nervous system first. After Tuesday, are you going to need to know these glial cells? Yes. Yes. Okay. And are you going to need to know where they're found and what their special functions are? Yes. Good. Good. Okay. That's exactly right. Why do I want to teach them to you by looking at this picture? Why? And obviously it's an illustration, but because looking at their structure, can it help us to see what their function? Can it help us to see some of what their function might be? Because they look different, don't they? All right, so I want you to tell me first, and they're labeled, but anyway, these are the neurons, aren't they? The big old neurons that are getting all the glory. And I make it sound like they shouldn't get the glory. They should get the glory. Okay, they get the glory. But they can't really function without these glial cells. So we have to really remember that. And what I want to talk to you about first, the first one I want to talk to you about is this one called the oligodendrocytes. These oligodendrocytes. Now, you've got this in your PowerPoint and in your text written. But these oligodendrocytes, I want you to see, look what this thing is doing. Here's one. You see the big nucleus. Do y'all see that? And is this another one? And is this another one? So y'all are seeing these. And what does it look like? This is the big nucleus, but it looks like it's sending part of its membrane out. And it looks like it's doing what to this neuron? It looks like it's wrapping. And what is the structure here on this neuron? What is this called? This is an axon. This is the axon. And it looks like it, and then it's sending another little thing out and wrapping here. And look, this one is wrapping here, isn't it? So it's just really, this one's wrapping then, you know, so you see the, what did you all tell me that plasma membranes are made out of? We can't forget what we learned, the lipids, which are what? Fats. Plasma membranes are 90% fat, aren't they? They are. They're 90% fat, most of that being phospholipids. So this membrane that's wrapping, and what you can't see from this illustration, but maybe you'll take my word for it, is that this didn't just send out an axon. Here's an axon from the neuron body. It doesn't just wrap it once. What this did was it wraps it up to 100 times. And it's wrapped. And it's fat. It's fat. I want you all to know the oligodendrocytes wrap axons of the neurons. And that wrapping, that membrane is called myelin. M-Y-E-L-I-N. Myelin. Myelin. So these oligodendrocytes are forming a myelin sheath of the axon. You see that? Now, why do you think, what function do you think that could serve? Protection. So you think that that's kind of protecting, kind of insulating, kind of protecting. Well, I don't want to disregard that function because that is true. But that isn't the main function. And here's what I need y'all to know is the main function. I need you to know that this myelin, this fat, allows for the signals to be sent quickly. So it speeds the signals that are being sent. Maya, you told me it took a microsecond for you to have all those images, those memories and those images flooding the frontal lobe of your brain, didn't you? And for that to happen that quickly, those axons had better be myelinated, because when the action potential is generated, that electrical signal can be sent quickly, 100 times faster, 100 times faster than if an axon is not myelinated. That's really much faster, isn't it? So you're talking about, because, and this is the way they measured this, you're talking about being able to go a yard or 100 yards in the same amount of time, right? Think of it that way. So myelin, which is a sheath that is mostly, what category did we say it mostly is? Fat, lipids, is speeding the signals that are sent along the central nervous system. And they better be, your central nervous system better be myelinated. When is this myelin happening? During development, during embryonic development. We started as one cell, a zygote. Then a blastocyst, which is a fluid-filled sac of cells, right? 2 weeks in a uterus, not outside of the uterus, but in a uterus is an embryo. Right? When did the myelinization of these axons, these neurons, happen? When does it happen? At the biggest rate. In embryonic development, all the way through infancy, but mostly in that embryonic fetal development is planned pregnancy is important. Can you make it up later if an individual, a child, has not gotten what they needed in utero? Is there any way of really truly catching up? There's some people who say that, you know, you kind of are who you are. And what they're really talking about are, this is an old wives' tale, but this is observations that people have made for years and years and years, millennial. They say you are who you are by the time you're 6. Well, guess what has happened mostly by that time? Myelinization. People taking care of making sure that they understand the importance of this. If somebody has breast milk that's allowed to sit out at room temperature, does anybody know what it looks like after it's set out at room temperature? It's more than half fat. Is that a good thing? That's a perfect thing, because what is happening early on during, you know, especially toddler, but even through the teens, what's happening? Myelinization of these axons in the central nervous system. Do you get it back? No. What is the disease where the myelin starts getting attacked? Does anybody know? Are they oligodendrocytes? Multiple sclerosis is when the myelin is getting attacked. All right, so oligodendrocytes, do they serve a really important function in the central nervous system? What are they providing? Myelin, right? Which is a wrapping of the axon. It is predominantly fat. And what is the function of myelin? To speed the signals that are being sent. If we were to, today is the year anniversary of what happened down in Florida, the massacre. If we were to hear, you know, but if we were to hear a gunshot, would it take us long to react? It would not take you long to react. Why not? Because of our myelin sheathing. And it would not take us long to react. It doesn't take us long to get to memories because of myelin sheathing. It doesn't take us long to be able to survive our environments on a daily, you know, even little whatever basis because of myelin sheathing, right? Okay, I thought of something that y'all don't want to hear about, but okay, that's fine. All right, so that's the oligodendrocytes. Now, I also want to tell you about these ependymal cells. So let's do these next. These are glial cells. And what do you all notice about these cells Are they closely packed together? Yes. Ooh, they are closely packed together. Yes, they are. They're derived of what type of cell tissue? Yeah, there you go. And let me tell you what they're doing. They're lining the central nervous system, and they're secreting. Oh, wait a minute. Lining and secreting. These ependymal glial cells are secreting cerebrospinal fluid. And cerebrospinal fluid is going to cleanse and keep the chemical stability of these neurons and glial cells what it should be. So is that going to be important? You secrete a certain amount of cerebrospinal fluid every day, and it's going through your brain and spinal cord, rinsing and keeping it where it needs to be, getting rid of things that it needs to, and you're reabsorbing a certain amount every day. That better be balanced. Mm-hmm. Yes, and it has to be balanced. You know, they say that there's a saying about cerebrospinal fluid floating in your brain. Well, guess what? It is floating in your brain, and that actually is protective, because it's protective in more ways than just chemical stability. It is protecting because it really truly is insulating. You know, so you have to have just the right amount, and you can't have too much, because if you have too much, it can put pressure on your brain. If we have too much, what do we call that? Hydrocephaly. Did you say that? Well, hydrocephaly. Hydro meaning what? Water. Cephaly meaning on the brain, right? So hydrocephaly. We're talking about normal. So we got to have a normal amount, and we've got to have this lining. This actually is nice. It's ependymal cells. Now, I also want you to see these little cells called, that's the second cell, glial cell. I want you to see the third one called microglial cells. Microglial cells. These are derived from white blood cells. Microglial cells are derived from white blood cells. And white blood cells, we know, do what? They protect us, don't they? So these are, they are your traditional white blood cells, but they're derived from white blood cells, and they are circulating around, and they are protected. So that's what their function is. That is what their function is. Now, I've got the last of the central nervous system glial cells I want you to know about. You ready? They are called astrocytes. They're called astrocytes. And tell me what they're doing. So look at this little illustration and tell me what it looks like they're doing. Would you say, Ashley?
Holding a neuron and the capillary together.
So it kind of looks like they're holding them together. But you know, that's one way of looking at it. But what's another way of looking at it, what it might be doing? Keeping them apart. Yeah, it's, you know, so isn't it funny? Like different ways, because it does look like they may be connecting them. But another way to look at it is they're kind of like keeping them apart, right? And actually, let me tell you about these astrocytes. The main function of an astrocyte is they have a lot. They're the most numerous, and they have the most functions that we know. Let me tell you kind of what they're doing. They're really separating the neurons from the blood capillaries. They provide what's called the blood-brain barrier. And you can see they're kind of looking like a barrier between these, aren't they? They provide the blood-brain barrier. They're so important. I'm going to go back to the text part here just to make sure that y'all see you've got it on your PowerPoint too, but you'll see it in your book. Some of you are going with your book. So they're the most abundant. That wouldn't be the most important thing to know about it. I don't think I even asked you that. But I want you to know their functions. They provide the blood-brain barrier. What are you talking about? Why do we have to have a barrier between blood? Our blood vessels are important because they're delivering at this capillary level. They're delivering to our tissues all the nutrients that we need for our tissues to survive, and they're picking up the waste products, and they're delivering fluids that can help the baby. So why do we need a barrier between our central nervous system tissue, brain and spinal cord, and the blood? What are you talking about? Toxins that could be where?
That could be in the blood. That could be fed towards the brain.
So you're right. This is right. You're right on the money there. Now, when we think about blood, we think of it being a wonderful thing, whole blood being a wonderful thing. And it is. It is a wonderful thing. But can you have transient bad things in your blood? Like, can you have transient, even if you floss your teeth too hard, can something get into your blood, and can it be moving around for just a short period of time? How about if you stump your toe? How about if you have a virus and you're viremic? Do you want that going to your brain and spinal cord? No, you don't want that being able to cross over. So you actually do have these astrocytes that are barriers between the blood and the brain. So nutrients can get across and waste can be picked up. But it's really tough because of these astrocytes. But that's a good thing. It's a catch-22 because it's a really great thing. It means that not just because something's in your blood, like maybe certain drugs that you're doing it to, maybe you should know you shouldn't be doing. You don't want them to get straight into your brain because it would be equal instant death if that actually happened. So there's some things that should never get to that brain and spinal cord, and it won't because you have the blood-brain barrier. But on the other hand, it's a catch-22 because when we have central nervous system diseases and disorders, do we have a difficult time sometimes getting across that blood-brain barrier with meds? We definitely do. We definitely do. An example of that is like Parkinson's disease. Has anybody ever heard of Parkinson's disease? And Parkinson's disease is a disease deep within the brain at an area, you don't have to write this down now, we're going to see in a second. But it's of the substantia nigra, which is deep in the brain, and it has to do with dopamine receptors. And dopamine is, in this particular area of the brain, is supposed to prevent unwanted motor contractions, but muscle motor contractions. But if you don't have enough of it there, then you get these contractions. And this is called Parkinson's disease. The problem is we can't get that across the blood-brain barrier. So they have to give something called a precursor, which is a much smaller molecule called L-DOPA. Maybe you've heard of L-DOPA. And that can cross the blood-brain barrier. Let me tell you the catch of that. We're just finding out in the last few years that accelerates the disease, the progression of the disease. I can see that. The problem is that it's still the best chemotherapeutic, so it's still being used even though we know that. So there are other things on the horizon for Parkinson's, and hopefully in the very near future, I think in the next decade, medicine will be almost recognizable from what we're doing today. That's how quickly things are happening in medicine. So that's why I'm so in awe of anybody choosing medicine as a field, because you guys, you're going to get to see so much and always be learning and always be, adapting to the newest things that are out there. But anyway, the blood-brain barrier, catch-22, because it protects, it needs to happen. If any of y'all have ever even seen on Discovery Channel, or you can Google nowadays and see brain surgeries, it's not bloody, is it? It's not bloody at all. As soon as you remove the skull cap and some of the superficial linings in the brain, it's not bloody looking at all because it's nervous tissue. It's nervous tissue. Those are the glial cells and the neurons, but it's really not bloody as long as you can avoid the vascular part of it. But those astrocytes are doing a great job. Now, I also need you to know something else. These cells secrete something called nerve growth factor, and I need you to know that because look what this does. What does nerve growth factor do? It is going to be required for the health of the synapses. It keeps the synapses clear. And must the synapses be clear? Yeah. Keeps the synapses clear, and it promotes the health of those. So this is going to be important. Now, when these synapses, when this is not enough being produced, then you can get what's called plaque formations at these synaptic areas. And these plaque formations won't allow the neurotransmitters to cross the space to talk to the next one. So you know what? I see that this is a pin. I don't know. I see it's a pin, and I can maybe even get to that it's a marker. But if I can't get to the cellular neuron that's going to send the signal to the area of my brain that lets me know what it's for, I'm going to get confused about what it's for. I can see that these are keys, but I can't get to the area of the brain that tells me what keys are for. I'm in my house I've been in for 25 years, and I can't remember which direction to go to get to the bathroom. What is that called? Alzheimer's. Because these plaques are forming in the synapses, and the synapses have to be clear so communication is getting to where it needs to be, to get to memories, to get to information about how to use something as simple, you know, as keys or a pen, or even remembering how to chew before you swallow, right? So yeah. So Alzheimer's. Which cells do we know are affected by this? The astrocytes. Okay, so do we have 4 glial cells that we just found out are incredibly important in supporting the neurons of the central nervous system? Are you all going to be able to match their functions to them? Yes, you're going to have to, not on Tuesday, are you? But by Thursday? Yep, by Thursday, because you've got these notes. There's 2 glial cells that are in the peripheral nervous system. There are 2 glial cells. They're called Schwann cells and satellite cells. But I would want you to know, and I'll just tell them to you now, so I don't know where the notes are, but there'll be some. The Schwann cells in the peripheral nervous system serve the same function that the oligodendrocytes do in the central nervous system. And what did the oligodendrocytes do in the central nervous system? Myelin sheathing. So the Schwann cells provide the myelin sheathing. If you all were to go back to your muscles pictures, and oh, sorry about that. If you all, see, can we call that Alzheimer's? If you all were to go back to your muscle sheet, your muscle innervation, you would see the end of that axon talking to the skeletal muscle. And if you look at the end of that axon, you'll see something that looks like it's wrapping it, a Schwann cell. And again, what does that help to do? Speeds the signals. Speeds the signals, doesn't it? The Schwann cells. If you've ever had, we can talk about this with, like, people usually know what you mean when you say sharp pain versus like achy pain. You know what I'm saying? If you ever had a sharp pain, those were myelinated axons. If you ever had sort of a dull, not really sure, kind of a dull pain, guess what kind of axons? Unmyelinated axons. Okay, so myelin, so Schwann cells. Now, the satellite cells in the peripheral nervous system, we're really not sure exactly what their functions are. Does that mean they don't have a function? That means we're not sure what they are. Okay, that's all that means. And by the way, and you also know that we're always discovering things that we knew about, like cells that we knew about, that we knew certain functions, but we're discovering new ones that we didn't know about, right? So these are the things that we know now about these. I've told you that. So are we good? This is a Schwann cell. A Schwann cell does this a little bit different than the oligodendrocytes do, because in the oligodendrocytes in the central nervous system, they were sending out appendages and wrapping, weren't they? But the Schwann cells in the peripheral nervous system, it's a cell that wraps. And again, it can wrap up to 100 times. That's a pretty thick wrapping, isn't it? And you can see how it begins and how it's doing, and it's happening at an incredibly fast pace when people are young. If somebody took 2 nerves and cut them transversely, right, which you all know means across, if they cut them transversely, look how thick the myelin sheathing is on this myelinated neuron versus this one that's not. That's kind of crazy, isn't it, how thick that is? So it really serves a very important function, where the oligodendrocytes kind of did it this way. The function of them is speeding the signal, which is impressive. Now, peripheral nerves. Did I tell you if it's a peripheral nerve damage, you might have a chance of getting it back, regenerating? You might not, but you might get it back. So they usually tell you that you would have it back in 6 months after the damage if you're going to get it back. But I think sometimes neurologists will tell people a year. But usually it's within 6 months if you're ever going to really get it back. But depending on the extent of the damage and depending on what's going on, you can get some regeneration of the peripheral nerves. Can you get them back from in the central nervous system? No, we don't think about it in the central nervous system. So nerve growth factor, we had said, is going to really be important too in keeping the health of the synapses clear. So again, when I talk about action potentials and resting membrane potentials, we're thinking about what's outside of the cell, extracellular fluid, compared to the intracellular inside the cells. We've got these membranes with very specialized receptors. Is that right? And we understand that neurotransmitters are going to have to cross this space, talk with, interact with, react with one of these receptors for the receptors to open. Is that correct? And then for this action potential to be generated, it is a millivoltage change in the resting membrane of the potential. It's a shock that happens when this happens. And this shock is going to be what talks to that particular cell. And whatever that cell is supposed to release at that other end of the axon will be depending on whatever the reaction is. I want you to think about this now. There's just something else I want to talk to you about. And then I think I'm going to give you all this extra time to start to do with the phones. But I want you to really be thinking about this. Our cells are specialized. Whatever the cell is supposed to receive, protein receptors that are supposed to be out there to receive, then you need to have enough of them, and you need to have them functioning, right? We said that the proteins that we make are dictated by our DNA. Isn't that right? But they're also dictated about what's happening in our environment. So would it make sense to you that, and this is something called, and you don't have to write this down, I just want you to be able to intuitively think about this, that makes sense. Does it make sense to you that, and it's called upregulation and downregulation, but does it make sense to you that if a cell, depending on how it was supposed to respond, if it received a neurotransmitter, the cell would get the signal and then place other receptors there so that it could be fully functioning and that go into what's called upregulation. And then there needs to be a period of time where these receptors start getting pulled off of that top surface of the membrane for downregulation, right? Meaning that the cell is slowing down. Are we good with that? All right. And this is something that we know happens. This happens with so many of the different neurotransmitters and hormones. Happens with hormones and neurotransmitters. Some hormones act as neurotransmitters and vice versa. But all I'm saying when I say that is that there's a chemical out here that can react with a protein receptor on the membrane, causing an action potential in that cell. Have I said anything tricky so far? All right. So this is why, and this is what I want to give you a couple of different examples. I want to give you a couple of different examples. One with the endocrine system and one with the nervous system. So is everybody ready to listen to this? And you don't have to write it down. I just want you to be thinking that this is happening in your system all the time. When someone says that your brain chemistry is off, has anybody ever heard that? Like the brain chemistry is off? You're hearing about it a lot now with kids and screen time. Is there any amount of safe screen time for kids do we know of now? No, because it is changing their brain chemistry. And what that means is they have these neurons that are flooding these synapses, causing cells to go into upregulation and sometimes staying in it too long. You get what I'm saying? So, and that is affecting the growth of this. So, and how the cells respond. So this is what this means. And we're just on the beginning of understanding this. We're already beginning to understand it. So thank goodness education is getting out there about this, because it's really important. And it doesn't just happen in kids, it's happening in us adults as well. But it's changing brain chemistry. And it's affecting how people feel and how people can function. So let me just give you an example of an exaggerated example, but I want to give you an example. I want to give you an example of dopamine. Dopamine is a natural, and don't write any of this down, this is just for you to sort of understand that we are ourselves and how our cells communicate. Dopamine is a neurotransmitter, just like acetylcholine was a neurotransmitter. But dopamine has its own unique chemical structural formula. And dopamine gets released in the brain, throughout the brain, but in the areas of the brain that we call our reward centers of our brains. And dopamine gets released and acts on the postsynaptic cell. And that postsynaptic cell starts sending its signals. And typically we think about dopamine as our feel-good neurotransmitter. Now, in the substantia nigra, it has a totally different function. It prevents unwanted motor contractions. But in the areas of our brain for reward and whatever, it's a feel-good neurotransmitter. And so we know, because we can see this through studies of what's happening in the brain, that when somebody is happy, when somebody laughs at a joke, or somebody sees a face that they know, and they see a face that they know, dopamine gets pushed out, right? Or has a good meal. Okay, so dopamine is being released, right? Sex, dopamine, right? So this is our, if you ever felt good about something, dopamine. Cocaine. Cocaine has a very similar structure, almost exactly the same. You can look it up as dopamine. So when somebody does cocaine, those postsynaptic cells, they get flooded, and those cells will push even more receptors. So not just, lasagna doesn't just taste good, it tastes great. The sex isn't just good, it's great. The joke wasn't funny, it was hysterical. This heightened thing. But the problem is this. What did we talk about was staying in a state of well-being? You've got to, even too much of a good thing is a bad thing. Because what happens is this. When you get this upregulation at that exaggerated amount, this postsynaptic cell is going to start getting signals to pull those receptors. And they start pulling them out. So then you get this crash kind of feeling, but that's not even the problem. The crash isn't the problem. And some of y'all are thinking, yeah, it is, because yeah, it is. No, it isn't. Because in a normal situation, your presynaptic cell would just get the signal that you've gone into downregulation, and your presynaptic cell would release some more dopamine. But when there's been this exaggerated response, the presynaptic cell is getting a signal to stop producing dopamine. So now you can't even feel normal. To feel normal, you have to chase the drug. Do you get what I'm saying? So really, it's one of the saddest things you'll ever hear, because this is truly at a physiological cellular level that we know this happens. And it's because of that stimulation that is affecting not just the postsynaptic cell, but also the functioning of the presynaptic cell. Do you hear what I'm saying? So that is a serious, serious issue. And some people are so sensitive to it that even after the first time of use, their chemistry is so sensitive to it that they think they're chasing a high, but they're not chasing a high, because it's changed their chemistry so much on the postsynaptic cell after the first time. They're chasing normal. And if that isn't the saddest thing you've ever heard, you weren't listening. So I want to tell you another situation outside of the central nervous system. This is with insulin, which is a hormone. But people who are producing tons of insulin because of, we produce insulin in response to carbohydrates. And so people who have had either, whatever, they produce so much insulin out here, the postsynaptic cell is going to get so tired of responding to it that the postsynaptic cells are going to be pulling their receptors off. And as they're pulling the receptors off, people who have type 2 diabetes, it's not that they're not producing insulin, is that their cells that are supposed to be responding have become insensitive to it because it was too much of it for too long a period of time, and the cells are now responding by pulling the receptors or changing the receptors enough that it's just not letting them in anymore. It's sort of like that neighbor that always knocks on your door. I guess, no, I'm not answering it. No, not really. I've never done that, actually. But I truly haven't. I was just making that analogy. Like, I'm embarrassed now because you think I have. I really haven't ever done that. But you know what I'm saying. That's kind of how the cells are responding. They're like, no. And it changes their response. So guys, so when we think about physiology, we have to understand it at a cellular level, don't we? And we talk about changing brain chemistry. This is at a cellular level. I haven't mentioned serotonin yet, but serotonin is it's a neurotransmitter, just like dopamine and acetylcholine are. Serotonin, though, is the neurotransmitter that some people associate with feeling good. You're wrong. It's not. Serotonin is that neurotransmitter that's just going to make you want, you get up and it's not like you want to be happy about it, but you're going to brush your child's hair because it's a rat nest, right? And you're going to brush your teeth because they just don't feel good. Your mouth doesn't feel good. So you're going to brush your teeth and then you're going to think, I'm hungry, so I'm going to go get something to eat, right? So serotonin is a functioning neurotransmitter. We need it to function. We secrete serotonin as neurotransmitters in our central nervous system by light clock and a 24-hour cycle, depending on sunlight. That's how we secrete it. That's why we're supposed to see the sun come up every morning. If you see the sun come up every morning and you go to sleep when the sun goes down at night, your serotonin levels have a chance of being where they need to be because it's based on exposure to light. What do we know about humans right now as far as sunlight goes and exposure? We're not getting it. What are our school systems that are supposed to have educated people on the school board? What are they doing to our teenagers daytime? They're saying, oh, well, they're really tired, so let's just start school late. That is absolutely the opposite of what we should be doing. They should be going to bed when the sun goes down, because when the sun goes down, that serotonin that you've had all day is changing. The molecule doesn't last forever. It's changing to melatonin so that you're feeling tired. And you're supposed to sleep at night because we step off of cliffs at night because we can't see. And we can't run from the bears because we can't see them, right? So we can't see at night. We're supposed to be asleep. And sleep is vital to the rest of the neurochemistry that's happening. How many of you know kids that they call it a witching hour between 5 and 6 at night? This is called a witching hour. And then how many of you know people who say, well, my kids, they just won't go to bed. That's because you didn't put them to bed as the sun was going down, that witching hour. And guess what their poor little sympathetic nervous systems told them? That they were in a fight or flight, and they start pumping out these chemicals because they thought they needed to be staying awake, and now you can't get them to bed. Guess whose fault that is? Not the kids. Kids are to go to bed when the sun goes down. And if you can get them on that pattern, they'll sleep until the sun comes up. Watch them. And then they should be outside when that sun's coming up. Not digging ditches the way my dad thought, but they should be, not if it's a tobacco field or digging ditches, but are picking up rocks. How many of you had to pick up rocks growing up?
As a
I know.
punishment.
So, no, not as a punishment, just because they saw you sitting doing something. I don't know, free. You were just vibing on this. So anyway, no, I'm just kidding. So, no, we really did have to clear our fields. But seriously, we are supposed to do that because that gets our serotonin levels. Y'all hear about serotonin and there not being enough of it as a depression, don't you? You hear about people who are depressed taking things like Prozac and other, so many of them now, that are serotonin reuptake inhibitors, meaning it's going to keep serotonin out here. All that means is it's going to keep serotonin out here a little bit longer. But is that going to throw something else off? Yes. Yeah, it is. And so you have to be careful with those I'm not saying people shouldn't be doing antidepressants. I'm just saying that we understand it. Here's the thing that would take longer, and that's that getting rid of electrical lights, trying to sleep at night, getting up with the sun, being out enough. We're at vitamin D deficiencies. This entire country's deficient in vitamin D. Why? No sun, right? But y'all hear when I'm saying at a cellular level, this is happening. And we'll talk more about serotonin, dopamine, acetylcholine, because acetylcholine, they call cholinergic neurons, are the ones that are affected in Alzheimer's, where the synapses are forming plaques. So there are, I introduced neurotransmitters to you with skeletal muscle, but there are a lot of neurotransmitters, more than 100 different ones known. You don't have to learn those, but we're going to learn about some of the common ones, right? And what they do. So how do you feel about the nervous system? Pretty good? So it's a little introduction, and actually that's a whole lot. I didn't even expect to get through that much today, but okay, we'll stop right here.